So thanks everyone for joining. This is our 22nd meeting, which is kind of crazy because we were started in 2017. We survived the pandemic, and here we are, 22nd meeting, the first meeting of 2024. Thanks everyone for joining. Just a couple of things. So this is our board, a lot of familiar faces. I'm the president. Rick over here is the vice president. Jackie is the secretary. David is stepping into a new role as the treasurer. And then we have four directors, Blake, Joshua, Casey, and Samara. And we also have two other officers. We have Michael Sezzi, who's our club conservator, and we have Hannah Roberts, who is our creative officer. And if you have noticed anything about our new website being revamped or our new Instagram, or all of the Facebook posts that have been coming out, we really have Hannah to thank for all of that wonderful work. So just a brief schedule for today. After this, we'll be hearing from three guest speakers. First off will be Diflora, who's actually coming to us all the way from Italy. So thank you very much for that. And then we'll follow with Craigsc Carnivorous Plants. They're a nursery in Virginia. And then we have Carnivero in Texas. We're very excited that, we've been talking for many, many years about going to the Pine Barrens. So we will finally be doing it this year. So Jason Ksepka, actually a previous steering committee member, will be leading that field trip, and he'll be telling you a little bit about what you can expect to see this year. We'll close off kind of by talking about our New Year's photo contest. We'll be showing you the results of that. And then we'll end by having a general Q&A session, mingling with all of you guys. And if you have a favorite carnivorous plant that you would like to show off, this would be a great meeting to kind of talk about yourself, talk about your favorite plant, why it's your favorite plant. Upcoming events for 2024, there's actually a lot of them. So in addition to meetings, we have a bunch of other invited events that we'll be going to. Coming up with March 9th, we'll actually be invited to It's a Bug's World, which is in Morgantown, West Virginia. And then later in April, there is a native plant swap and sale that's closer to the Norristown, Eagleville area in Pennsylvania, if you know where that is. June 1st, David Wallace, our treasurer, will be leading a bog pot workshop at Black Moshannon State Park. That was actually the location of our field trip last year. It's a beautiful place, beautiful park. You can see a lot of carnivorous plants in the wild over there. June 22nd will be our 23rd meeting, and it'll be the field trip for 2024. We are going to the Pine Barrens. September 28th, we'll be participating at the Hardy Plant Society pop-up. That's in Collegeville, Pennsylvania. October 4th through 6th will be Keystone Clash in Morgantown. This is an event that we've been participating in for the last couple of years. And then also in October, we're still working out the details, but our 24th meeting will potentially be at multiple sites from Washington, D.C. all the way through Virginia. And then we're also really excited to announce a new members initiative. Rick will be talking a little bit more about that later today. His initiative is MACPS Connect, and it'll be a monthly event starting every third Saturday of the month, starting March 23rd. So definitely stick around to hear about that. It'll be a lot of fun. I want to talk a little bit about membership. So membership structure has generally not changed, although hopefully we've made a bunch of improvements to actually make it a little bit more streamlined, and so you know a little bit more about what you're actually getting for membership. So if you do become a paid member of MACPS, you're allowed to sell in our plant auctions. 20% of the proceeds do go back to MACPS. You'll get increased access to any prizes and raffles that we do. You also have a 10% discount in Carnivorous Plant Nursery in Maryland. And then, as I mentioned earlier, and we'll be talking a little bit more about that, there will be an exclusive monthly member live stream, also if you are a paid member. You can see the prices of our memberships below. Individual memberships, they start at $20, but we would welcome donations up to $40. Family memberships start at $30 and go up to $50. Student memberships are $5 a year, and corporate memberships are $200 a year. As always, visit our website, macpsociety.com, to join. You can submit a form right there, and then also submit a PayPal with the amount. Membership lasts 1 year from payment date. So for example, if you buy a membership today, your membership will expire March 1st in 2025. So thanks for coming. If you haven't joined if you haven't followed us on Instagram, please do so. We're just starting this off. Hopefully we'll have a pretty nice social media presence, and thanks to Hannah for creating all of this. So at this point, I'll stop sharing, and I would love to invite Valerio of Diflora to give us a talk. We're super excited to have him. I have personally bought plants from him. They have been in fantastic shape. I'm super excited about all the stuff that I've gotten from him, and I'm really excited to hear from him too. So thanks.
Hello to everyone. Can you hear me?
Yes.
Oh, yeah.
Okay, perfect. So, hello to everyone. Thank you, Kevin, for inviting me at this event. First of all, I'm sorry for my quite poor English, but I will try to do my best for you and for you as people there. You should be lucky to have people like Kevin managing so many events, because here in Italy, we don't have such an active association managing all these events all during the year with field trip and so on. So you are lucky enough for having this association there, and I'm happy to be here speaking a little bit to you about Diflora. Kevin, I don't know how you want to manage this speaking. So if you prefer to have a presentation by myself or something like question and answer, let me know how do you prefer to manage the speech. So should I do a presentation for first?
It's whatever you like. I think a presentation, maybe like a brief one, could be helpful, but it's really up to you. We're just excited to have you.
Okay. So as you can see, we are in the main in vitro chamber of Diflora. So this is our in vitro grow room, half of that, to be honest. And so Diflora is a company based on micropropagation that started 10 years ago, something like that, like an hobby. So it became professional in 2019. So few years before pandemic, COVID pandemic. And so we started as an hobbyist, and we started building a very, very simple homemade laminar flow hood, and starting experimenting with a lot of carnivorous plants. And everything became more and more interesting for customer and for people all over the Europe, but also outside Europe. And so we decided to start a full job, and we invest in some more professional equipment, more professional structure. And so after 5 years of after becoming a professional company in 5 years, we are here. And so we are very happy of what we were able to build. Diflora starts with the passion of tissue culture and carnivorous plants together. We were able to merge these two passions, and little by little, we start growing up our knowledge and know-how about in vitro introduction, in vitro propagation, and also greenhouse cultivation of carnivorous plants. And we have two facilities, one in which our employees will manage all the manual work. So laminar tissue culture, plant transfer, and tissue culture media production. And another facility here in which we have the in vitro grow chamber, an acclimatization grow chamber for acclimating, so adapting the plants from the controlled condition inside an in vitro tissue culture vessel to the stronger and harder condition of the greenhouse life. And also a shipping area in which we process all pack and everything else related to shipping. Of course, outside this building, we have also something like a 1,000 square meter greenhouse in which we have all the standard greenhouse part in which our plants are growing as small plants or potted plants. So this is how our facilities are structured at the moment. And here we are inside the in vitro grow chamber, in which at the moment there are mainly Sarracenia, Venus flytrap, and Rosella. Some curiosity for you, for example, we have had some massive contamination 2 years ago. And so we started almost again from conservation flask something like 2 years ago. And we have some delay with Nepenthes, Darlingtonia, Cephalotus tissue culture, because as Craig can confirm later, they are very, very slow grower in vitro. And so we are still ending the in vitro propagation plants, waiting for their acclimatization. So at the moment, we have been able to recover from this massive contamination problem in vitro. And we have been able to acclimate almost all genera, apart from Nepenthes and very slow grower ones. And Yeah, why I'm talking about contamination? Because as you probably know, tissue culture is made using sterile environment. So we are growing plants inside this sterile container in which an artificial medium composed by salts, gelling agents, water, and sugar allow plants to grow, to multiply, to stimulate root formation, and all other morphologic aspects of plants. In this environment, plants are able to grow faster, and we are able to control a little bit more which type of stimulus we would like to give to plants. So, as I already told you, something like multiplication, growth or rooting of plants, we are able to manage and control those processes a little bit more using phytohormones or using tissue cultural media composition. In this aseptic environment, we are able, especially to induce multiplication of plants that need to be propagated as cultivars. So they need to stay, they need to be the same genotype of the parents because they are going to be sold for their feature. So I don't know, some Venus flytrap space werewolf or something like that need to keep their features also in the future generation. So, as you probably already know, starting for standard breeding, that is made by pollination using flower, pollen, and stigma, and everything else related, allow to produce seeds, but with seeds we have crossing over, and so the some produced by this crossing are all different and cannot be sold and spread as a real cultivar. And so tissue culture allow, starting from tissue culture plant, part, sorry, of the plant, so leaf, root, flower stalk, or flower itself, to induce new plant production from tissue, from the tissue of the plant. And this allow to keep the genetic of the son that is the same of the parent. So tissue culture is used for this purpose, so to produce a huge amount of tissue cultured plants that is genetically the same from the parent. So it can be sold like the parent's name. This is the main purpose of tissue culture. It can be achieved with a lot of different ways. And at this point, if you have any particular curiosity, don't be scared about asking anything you are curious about, because tissue culture is a very, very wide field and a very, very wide technique. So if we speak about something of your interest, it could be better. So feel free to ask anything you want. I can go on with the company description. So after this very, very complicated tissue culture part, as I already told you, we are able to acclimate everything in a grow chamber. I already told you about this grow chamber that is a vertical farm. So it's still inside the building, and we grow everything under artificial grow light, both temperate and tropical species, and with controlled temperature and humidity. These allow the first step of acclimatization. So of the adaptation of these plants to outside conditions. While I'm saying adaptation of these plants, because inside tissue culture vessel, first of all, plants grow with sugar. So they produce very, very few photosynthesis, and they use directly sugar for recovering energy. Standard plants use the CO2 for producing energy thanks to sun and thanks to photosynthesis. Of course, plants in tissue culture are hermetic and so are not able to have a lot of gas exchange. And so they use directly sugar that is present on the medium to produce energy. This is the first difference. The second difference between tissue culture plants and standard plants is the ability to grow with low humidity. In fact, inside this vessel, humidity is 100%. So that means that the stomata, I believe that is the right English word, I don't know, the stomata of the plants are already opened. So transpiration of the plants are always active, always 100%, and it allows to have a lot of that always opened stoma allow to have a lot of transpiration and allow the plants to collect a lot of mineral salts and nutrients from the medium. So the growing of the plants is 100%. Instead, outside and plants that live outside need to control this transpiration because they cannot always leave stomata open, but they need to control them. And so leaves that comes out from tissue culture vessel need to adapt. So need to build new leaves that are able to manage this closure, these open and closed stomata outside in the greenhouse. The in vitro tissue culture leaves are not able to manage this transpiration control. These are the two factors, two main reasons why plants need to adapt to outside conditions. If you place plants that comes from tissue culture vessel directly outside, it happens what we call burned leaves. So leaves become completely brown and die off. Maybe with Venus flytrap or Sorrentinia, the rhizome will survive and then the plant can sprout again. But with other plants, herbaceous plants like Drosera, the plant could die, especially if attacked by mold or other fungi. And so this is the reason why we have this grow chamber. And so here we have a temperature controlled chamber in which humidity are little by little decreased from 90% to 70, 65%. And the light intensity are little by little increased from something like 4,000 lux 12 hours per day to something like 5, 6, 7,000 lux 16 hours per day. During a period of something like 1 or a couple of months. After this period, plants are ready to be placed outside in the greenhouse under standard sunlight and standard greenhouse conditions. And so this is the reason why we have this indoor grow chamber and why I'm saying also temperate plants are placed inside for these 4-8 weeks. Because also temperate plants need this adaptation process. Also, if they are more tolerant to harsh and dry conditions than tropical plants. After this period, we place plants outside, of course, and we have a greenhouse that's a tropical greenhouse in which there are always shade, 50 or 70% of shade, and in which we have a nice rain system to keep, especially Nepenthes, but also Sphagnum moss, always with a high humidity, and a tropical greenhouse in which we have simply the standard tray, water on the tray method, with which probably every one of us grow standard carnivorous plants. The only difference, I don't know there in the US how is your summer, but here we have very, very sunny and warm summer, and so we need to shade 50% tropical temperate plants also during summer. Also if they love full sun, but here is too hot, and so we need to shade them something like 40-50% when temperature is above 30 degrees Celsius degrees, of course. And so I believe that this is the whole process with which we produce the plants. And since something like 2-3 years ago, we are able also to export plants directly there into the US because our company is certified to be an artificial propagator company. And so we have CITES and the phytosanitary certificate and the certification. I don't know why we need to have this CITES certification in Italy because we need to prove to the US that we are selling their plants that have not been collected and poached from nature, but the plants are native from the US and not from Italy. But CITES is an international rules, and so we must respect it, of course. Also, it sounds like a contradictory, but it doesn't matter. And so we are able to export directly plants there. And I will say nothing more from the production process. And the only thing that I'm very proud of is that we produce 100% of the plants we sell. So we start from a seed, from a flower start, for example, for Venus flytrap, or from a leaf, and we produce everything. So we buy only one mother plant from each type, and we produce everything else. We are also starting a way to mass propagating Sphagnum moss in vitro, with the aim of producing our own Sphagnum for growing purpose and to being able to remove peat from cultivation, and so to prevent as much as possible the poaching of peat for horticultural purpose. So this is the aim, our aim in the next 3 or 4 years. So to switch from peat-based growing media condition to Sphagnum-based growing media condition. I know that you're there in the US, you use way more than here in Italy, dry Sphagnum moss, but we need to do a lot of testing, especially with small plants that came out from tissue culture, because I believe that there are some differences between cultivation in peat and Sphagnum moss when talking about the young plants. And so, yeah, this is what a small resume of what we are doing here in Italy. And I would like to see some questions from your side, so we can optimize time, not speaking about anything in general, but speaking about something of your interest. I see that there are already a question on the chat. So if you want to ask me something on the chat, I will read them and try to answer to all questions. I read something like, can you speak about why lighting is important to use in tissue culture? Yeah, this is a very interesting question, because lighting in tissue culture is important mainly for photoperiod perception into the plant. So lighting is important for many reasons inside the plant. When we are considering plants grow outside in the greenhouse, is very important because of energy reason, for energy purpose, like we already discussed it, because of course plants need to fix carbonic gas on the atmosphere, so CO2, to produce their own sugar, that is the energy bricks every plant have, and then they use these sugars for producing energy. While in tissue culture, we have already sugar in the medium, like you said correctly. But light is important not only for this reason, so not only for energetic reason, but also for photoperiod perception. We know that a lot of stimulus inside plants are regulated by photoperiod. So flowering of Venus flytrap in spring is regulated because plants are able to perceive that the day of light, the hours of light during day is longer and longer with the spring that is approaching. For the same reason, for example, tuberous drosera sprouts out because they are able to perceive that the temperature is colder, but also because they are able to perceive that light is the hours of light is longer, and they go and they start to produce tuber because they perceive that the hours of light will become longer in spring, and so they start to go dormant and to produce tuber. For the same reason, plants love light in vitro, especially the 16 hours of light and 8 hours of darkness, because with this light, they are with this photoperiod, they are in the vegetative stage. So they produce every time new plants, and they multiply very, very fast. Growing plants in the dark will result in an etiolated plant, but also in a plant that don't know in which stage of his growing cycle they are. Another very quick thing, then is which question is that, for example, tuberose drosera prefer an in vitro grow light of 12 hours of light and 12 hours of darkness, because with 16 hours of light, they induce tuber formation every time, and it's very difficult to multiply them with a long light condition. Another question by Yaron, how do you deal with toxicity build up in tissue culture container, and do you offer phytosanitary certificates? Of course, we send everything out with phytosanitary certificates that stand also as a secret document. And so we, of course, export everything legally there. It's very, very easy for us. It's something we do since 3 years. Toxicity with in tissue culture containers. This is a very, very nice secret I'm going to share only with you. You should keep with you only, but Craig can also confirm it in its next meeting. When you're going to have a tissue culture container, let's see if I am able to find one. I don't know if I have something here around me that becomes brown. And so it's what you call probably tissue culture toxicity. It is because the container are sealed, are closed. And so ethylene, that is a gas produced by carnivorous plants and plants in general, are not able to spread out in the atmosphere, and the concentration increase little by little inside the container. And ethylene is a phytohormone that produce tissue death, so apoptosis inside plants. So if you switch from this container to vented container, you solve a lot of problems of tissue culture toxicity. Another question from Rick. How do you manage to source Nepenthes seed stock specifically? We have very, very few Nepenthes at the moment, and we are searching for more of them. Typically, we buy plants and we start in vitro propagation from tissue, so from leaves, from stems, and from part of the plant. It's very difficult to trust seed sources, especially because you know better than me that most of them comes from east of Europe, and you don't even know if they are really what they are selling. It's very, very difficult to know, to be sure that what they are selling is what they are declaring. That's the point. You're welcome, and I'm very glad that you received your answer. If you have any more questions, don't hesitate to tell me anything else you have in your mind, and specific about tissue culture, but also about carnivorous plants in general. We are doing a lot of research. We are doing a lot of experimentation with new breeding of Venus flytrap, but also some other plants. We are doing a lot of research on how to multiply in vitro very white variegated plants and pom-pom-like form of Venus flytrap, because not all the plants will keep its features using tissue culture organogenesis, so flower stalk or cutting propagation in vitro. White variegated plants and some form of pom-pom Venus flytrap are not able to keep its feature. Also, if we start from a tissue, so from a cell. And so it means that probably these mutations are not generated by a genetic base, by something that is not genetic, from RNA or something that is not in the DNA. Yeah, you are asking me some, what is challenge, we have few experience also with Elianthera, like with Nepenthes, because we have very, very few species. At the moment, we did not find difficult to propagate Elianthera in vitro. We did find only very low. While with Nepenthes, there are a lot of differences in tissue culture propagation in vitro. And so there are plants that are very, very easy to propagate, like lowland Nepenthes, and other ones that instead are very, very easy to propagate, very difficult and very slow grower in vitro, like the Highlands one, the Raja, the other Edwardsiana, and other famous species. With Elianthera, I see that the there are not so wide differences between species, but they are all quite slow grower and slow. They multiply quite slow in vitro. So I did not find it difficult, but slow at the moment. We only made that in vitro propagation, so I have never tried with them standard propagation technique, and so this is everything I can tell you at the moment. But we are propagating only minor nutants and few other hybrids. I find them very it's way more difficult to let them germinate in vitro because like also some type of nepenthes, they produce a very young sprout, but then it tends to die. We still need to try to understand why, but it is probably due to an excessive of nitrogen content inside the growing media, I believe. Probably the young plants are again more sensible to high nitrogen content more than mature plants. So I believe that our time is also coming to an end. We have another 5 minutes available if Kevin can confirm it. But if you have any more questions, if not, I'm going to a conclusion as you prefer. But if you have any more questions, I will be more than happy to answer to other curiosity.
Valerio, yeah, thank you very much. I'm just, always thinking about ways that we can get members here more plants. And so, I ordered from you a while ago. I don't know if this is still your policy, but I remember that you had a limitation on a number of plants that you could ship. I would love to get a group order together somehow. And I know shipping from Europe overseas often involves a lot of, just paperwork or a lot of costs. Is there anything that we could maybe do to, say, facilitate a group order here in the US?
Yeah, to be honest, we don't have anymore these plant limits because we are using now a partner there in the US. So we don't even need an import permit directly from you because we are using a partner there that is collaborating with us. And so we ship everything to him and then he sends everything to final customer. And so it becomes everything way more smoother for everyone. And so there aren't a real plant limits at the moment. Of course, there are these two possibilities. So to buy directly from us, but you need an import permit to overcome these 12 plant limits, or to use this partner that you don't need to have an import permit. You don't need to have anything at the moment. And there is also anymore the 12 plant limit we have in the past. And
Fantastic.
so it's very easy. You can simply buy from the website. We will manage the shipping to this import permit. We have a very nice agreement with him and also to the USDA, so to the plant inspection station of this partner. And so they are very smooth also on the custom clearance process because they know us, they know that we are working well. And so the whole process it would require something like 8, 10, or 12 days, no more, apart for some strike that sometimes happen for DHL there. Because, the 3-day parcel will be there. They are inspected in 1 or 2 days, then it's picked up by our partner, and the USPS internal shipping there takes something like from 2 to 4 days, so something like this. But we have solved all our past problem with this method at the moment.
Fantastic. Thank you. You have one more question in the chat, and I think we'll call it, if that's okay with you.
Yeah, of course, we ship plants by route because for US importing routes, we require that plants have no soil, of course, and also they are free of plant nematodes that are some parasites that live in the soil. And so we ship plants by route and with all papers needed. And in the last 1 year, we haven't had any problem and any stop also after plant inspection. So I believe that the process are quite tested at the moment and everything is working well.
Awesome. Well, I think that's all the time we have. Valeria, thank you so much for taking time out, especially on a Saturday evening for you. Everybody
Thank you
who
too.
haven't been to Diflora's website, like we put in the chat, please go check them out. They have some incredible varieties there. Thank you.
Thank you to you all, and I hope you enjoy your next meeting and the full this full call. I cannot be with you for the full 2 hours because it's dinner time here. But I hope you can enjoy also the other speaker after me, and say ciao to Craig and to Carnivero staff that I know them very well, and so say ciao by my side.
Thank you very much. Take care.
Ciao, ciao.
And then I'll leave it to Rick. I'll have Rick actually introduce Craig. Thanks.
Yeah, thanks, Kevin, and thanks, Valerio. So our next speaker is going to be Craig Heath of Craigsc Carnivorous Plants. He's based in Northern Virginia, and he specializes in primarily Venus flytraps, but he also does tissue culture with cephalotus and helianthera. I think he might also be branching out in a couple other things these days, if I'm not mistaken. Craig's also got an accomplishment of registering several cultivars with the ICPS, so you may be familiar with some of the popular creations that he's come up with for Venus flytraps. So I'm going to go ahead and turn things over to Craig if you're ready to go.
Let's start with I guess how I began and started to do carnivorous plants. I've been doing plants for about 15 years now, and I would buy them and then I would grow them and then I would divide them and I'd sell them. And most of my sales, I did a lot of them on eBay. So I decided to go into the tissue culture scene. I asked a few guys, especially Frank Trombone, big tissue culture guy that helped me a lot. Also, I bought the Carol Stiff, the kitchen culture kit, and that's what got me going. As I went ahead and did all that, I started out as an aquarium, 30 long aquarium on its side, and I had a light through the top of it, which I was doing all my sterile things and putting in baby food jars. As time went on, I did the B52, that did good. I did get some contamination, so if you decide to go that way, don't be discouraged. It's just the way things happen. You just keep plugging along. And when you get one strikes, then you're excited and you want to do more. From there, I moved over to a laminated flow. I wanted more room, less contamination. So I got a hold with Frank Trump, and he always says, bigger's always better. So in the background, as you can see, I did a 30 by 24. Built it all by hand. Those are expensive, but I started out as in alcohol with 4 steps, and you dip them into sterile water. From there, I was doing the pressure cooker scene, where I was using a 32 quart pressure cooker. I was doing baby food jars in there. Well, and I had a hot plate, but that was time consuming, and I wanted something that would be a little bit faster. So I moved over to the microwave, and I started doing tissue cultures in little deli containers. I did the 2, 4, and 5 ounce, and also moved up to the 16 to 32 ounce delis, which are microwavable and they won't disform. With the pressure cooker, you can only do so many of them, but it would take you 15 minutes to pressure cook, but to wait for time for it to heat up. And that's like, talking about an hour. Microwave, it was quick. Put my medium in there, leave the lid on, don't strap them. And once they start to boil, then I turn the microwave off and let them cool on the table. Once they cooled off, then I put them in a container, like a big Bambarine, couple of 70% out there, and then they close the lid on. Told me wait 3 days. Contamination pull them out. But I have really good luck with that. Now I don't have any contamination. My hood that I built was like in 2019. The only expensive part was the HEPA filter. And that can run about $200 for one. What else? As for media, like Barry always say, it's out of distilled water. It's got sugar and it's got vitamins in it. And either you want to put hormones in it, that's totally up to you. Now, the kitchen culture kit that I bought, I was always using, it's called a BAP. And that's for using to form calluses. Now, the NAA is the part where it tells the plant to start growing roots. There's all kinds of recipes out there for that. A lot of them, I deal with a lot of cephalotuses, some helianthores, the minors. I always kept those propagations going, and it's probably been 5 years, but I always keep the thing flowing. Always changing like the media out when it starts becoming brown or tint color. Then it's time for me to, the plant's telling me it needs to be replated into new media. It's usually like 3 months that you have to do that. So you have to watch those. A lot of my fly traps are done by rhizomes, stem cuttings, leaf pullings, and root pullings. A lot of my other plants through the International Carnivorous Plant Society have some that are registered. I believe it's the Destruction, Red Onyx. Can't think what the other ones were. I don't know. They're on there. It's CCCP, and you'll see them on there. A lot of my lights you can see in the background, they're all fluorescent. I had T8 bulbs, which are becoming obsolete now. And if you do get them, they're very expensive. They do run a lot of electric. That's why I went with LEDs. They're usually about 6,500 kWs. I do mix the bulbs with 65 and 3,000, trying to get like the growth and the flower producing spectrum colors. A lot of them are in 10 by 20 trays. Humidity zones, I do control them to a lot of my plants that do come out of the tissue, good luck bringing them into soil, and I do have them in sphagnous moss mixed with perlite. But in the meantime, when you do take them out of the tissue culture containers, you have to run them under warm water to get all the agar off the roots. I put them in like a little sieve, and I have a little spigot on my sink that it's like sprays on them, and I try to pull them apart, and then I put them in distilled water containers to let them soak for a little bit to make sure all the agar's off, and then I just transplant them into sphagnum moss to control the humidity to make them grow. Now, to understand why we want to acclimate them, the person told me it's like the plant doesn't have lungs. Without lungs, you can't live. It won't breathe. It won't grow. That's the simple term to understanding. It usually takes about a couple of weeks, and then I start giving them air a little bit, control them, watch them, make sure they don't create any mold, which some do, which is normal. So just keep plugging along and watch them. I use a lot of rainwater. The only time I use distilled water is when I make my media mix up. I do TDS it. Use the rainwater here in Virginia. It's less than I can get during the cold winter. You probably get 5. I do have a reverse osmosis. I run through a 55-gallon barrel, and that usually puts out 0. What else? I'm dabbling into the helis. I'm trying to understand how to propagate them. I do have a little bit of a mold problem, but I think I've got it cured. But I'll show you what mold looks like, or contamination. Let's see what that. It's a little hard to see. Let's see here. That's contamination. And the easy way to describe it to somebody, it's like bread being that has mold on it. It's like the microbes got in there. They love the sugar, and it just kills the whole plant. And there's nothing you can do about it. So it's basically trashed. I've done some fly traps before where I've probably pulled 5, 6, 7 cuttings, and they just won't strike or they get mold until you get one that you get lucky on, and then it's like, all right, you know. But the thing is, just if you want to dabble into it, just keep going at it. You'll understand it. Telling is a lot different than doing it. Once you do it on hands-on, you'll understand how it is easy to do, what things not to do. Like for me, I wear a lab coat, mask, rubber gloves, 70% alcohol, sterile wipes, all those little things like that all can come into consideration not to have contamination. A lot of people, like I said, Frank Trebone, big tissue culture guy, and he's on Facebook. I believe it's the Tissue Culture 2020. I believe he's on, but he's not the administrator anymore. I think he's kind of pulled back out of it, but good site to get onto. Good people. Another good guy. If you were into it, there's this chemical called ppm, and what it does is helps control the contamination into your media when you make it, mix it up. And that's through Francisco, good guy. He's got a website. It's called Plant Tissue Technology. Got videos on there. Great guy. Has schooling or online classes, and he always gets back with you really quick to if you have any questions or answers. So, but that's my life. I'm retired. I do this part-time. Now it's become full-time, and the family operated business. My wife does all the business work. And that's how it goes.
I have a question for you, Craig. I don't have a chance to ask this specifically of you yet, but when you finally get to the point where you've got something like destruction or red onyx or anything like that, what are the procedures and policies or steps that you have to take to actually get something registered with the ICPS officially?
With them, you have to fill out the form. They want to know where the parent plant came from, if it was mixed with something. You have to a brief description, how long you had it. You have to measure the fly trap. You have to measure the leaf cutting, or I'm sorry, not the cutting, but the leaf. You have to measure that in centimeters. That's all they want is centimeter stuff. They have to go through a person that has to review it, and then they usually takes a few months for them to get back to you. Either they want you to retweak it, and if it whatever it's similar with, or any other information that they need. Then you also got to send pictures too.
I've heard that there's a quantity element to it as well. Is that true, where you have to produce a certain number before it's eligible, or can you just have a single plant that can be registered?
I have like a single plant that I register.
But you don't have to make like 20 of them before.
Well, if that plant comes from the mother plant, then I can make more of that type.
Mm-hmm. So they don't require that, like, hey, in order for this to be a cultivar, you got to have 20 replicated destructions before we can say yes, it's a cultivar, or do you just have to prove that it's something unique itself and then that's enough?
Usually I start, when I do, like, say, destruction, I grow them out to make sure that they're stable, and then when I see that they are, then I'll just register that one.
Gotcha, understood. We do have another question in the chat.
Sure.
To know, where can we purchase plants from you?
Most of my plants I sell on eBay. Right now I have none on there right now, because it's cold here. I usually wait till it's a little bit warmer. And I have a Craigsc Carnivorous Plants on
Thank you very
yeah.
much.
So these,
Oh, sorry, I had a question, if that's okay. Um,
sure.
I was just wondering, for someone who doesn't do any tissue culture really, any tips just on how to get started, or kind of just do what you did, get the kits, try to design your own hood, things like that.
Right. If you go online, you could see how people don't have to. I've see people use like a plastic container, like a tub container, and they flip it upside down. They put holes in it, and then they just use 70% alcohol. They stick their arms in there, and that's how they do their tissue culturing. I think there's a woman called the Plant Jar Girl on YouTube. I think she does a lot of that, but that's the easiest way to do it. I've done it with a 30-gallon long aquarium. The face was open, had my light on top, wipe it all out with 70% alcohol. And what the thing is you don't want a whole lot of air movement going into the aquarium. And that's where you get your contamination. So you got to be really slow. You got to be careful that you can't cross your arms. That can drop chemical microbes in your contamination, or you can get contamination that way. Everything's got to be sterile. Forceps, 70% alcohol, sterile water, jars. And that's how I start.
Awesome, thank you.
And Craig, why don't you let the folks know if you're able to, what parts of the plants are you usually using when you're doing, fly traps or cephalotus or, anything else that you're working on, like sundews or whatever?
For my cephalotuses, I had bought a vial from a place over Czechoslovakia, and it was in a sterile container. And that's how I kept the culture alive. You can buy them from over there, but sometimes it takes forever. You get a little baby plant. Usually it takes about over a year to multiply them. Some grow very slow, like the cephalotuses do grow slow. As for the fly traps, most of my cuttings are from leaf. Once you sterilize them, which is you could use a bleach solution, which is, I believe that was the old thing to do. Now I believe it's too strong for the plant, even though you get like a baby leaf. That's what you want, a fresh, young leaf. And like I said, the Clorox, I believe it's too strong, and it'll kill it, and it won't produce, and it'll just turn brown. Other alternatives is pool chlorine. Less strong. There's a percentage, and you soak that for a certain amount of hours, and then you don't have to do any rinsing as you do when you're working with bleach. You have to rinse it, but with chlorine you don't. I cut the ends off of the fly trap leaf, and then I put it in the media. I don't have to rinse it. And then that's how I get mine to grow. And it's all give or take if it's going to strike or not. Like I said, you might do 5 or 6 pullings and you just get that one that works. For hydrogen peroxide, for seeds, I use Clorox. I use filter paper. I fold it to round paper. I fold it in half. I put my seeds in there, fold them up, put a little plastic clip on there, and I soak them in Clorox with a percentage of sterile water. Soak them for how many minutes? I think it's 7. Then I take them out and use sterilized tweezers and put 1 seed per container, and see if they take.
Do we have any other questions for Craig out there for the people that are tuned in? Feel free to pop them in the chat there, or we can definitely have them answer anything else. Otherwise, we may be moving on to the next folks.
Okay.
I do have another quick question. I think one of the things that is really exciting about tissue culture is it allows us to, put in the market plants that are very rare in cultivation. I was wondering if you have any exciting plants that you're hoping to propagate soon. Anything that we
Uh,
can look out for.
right now I'm working on the helium forest. That's a one thing that I cannot find answers about. I've got some rare ones that I'm working on. I'm having some little difficulty with contamination, but I think I got it solved. But like I said, they're very growth slowers. So I got my fingers crossed. I think somebody had a question about microwave. When I make my media up, I use like 5 ounce little containers, and they got to be I think it's PP5, which are polypropylene. They can withstand microwave. Um, when I make my media up, I put let's say a 5 ounce container. I put like 35 milliliters of media into it, and place them all in the microwave. Place the lids on top. I don't snap the lids. I just lay them on there, and I start the microwave up until the media just starts to boil, and then I stop, and then I pull them out, and then I put them on a table with the lids on top, and I let them cool down. If you don't let them cool down, the containers will crush in, and you'll have deformed containers. But you got to let them cool down. Once they cool down, you can snap the lids on them, and I put them in a container. As for vented lids, I understand that there's no problem with vented lids on the on your containers. Now, I haven't gotten into the bio couplers, but I know they have um, spore filters on there, and I understand you cannot get those wet. Most of my containers are not vented; they're all sealed, um, which they do pretty good. I was into the baby food jars, but you know, they get quite expensive, especially with the lids that come from Pyrotech Laboratories. They get a little expensive, and they get old after a while. When you microwave them or pressure cook it, they start to crack, and you just throw them out. So that's why I went with plastic containers. A lot of testing, you can see in the back here. I'm doing a lot of testing with chemicals, with NAA and BAP, certain percentages, trying to find out what's the sweet spot for my cephalotuses. Also, some other plants. I'm trying to see what grows good, what produces good. So those are my little experiments that I'm doing. Also, when you do tissue culture, always write your notes down. I've always had to go back and find out things that I need to know. That it's a big plus. Got to have your notes. Things take time. Got to be patient. Some things take over a year. Some take more than that. It's just all on how they genetically how they grow. Some grow slow. Some grow vibrant. All mine's. I have mine. They call it. Well, I call it the man cave. They're all in the basement. All my timers, like I said, the LED lights, they come on and off. And that is it.
Awesome. Thank you, Craig. I really appreciate you speaking about it and everything. And I definitely will certainly let folks know. Absolutely keep your eye out for his stuff as it comes out. You can buy, if I'm not mistaken, unless you change things, Craig, you can buy things in the flask if you want to try your hand at doing anything.
Yes.
Out of tissue culture directly, but I believe you also do hardened off plants as well,
Yes.
and in groups and whatnot.
Yeah.
We'll certainly do to keep an eye out for that, folks. He's got some good stuff.
Thank you.
Well, thank you, sir. We're going to go ahead. I believe Drew Martinez might be ready to go with as our next speaker. I'm not sure. I'm going to go ahead and turn things over to Kevin. Make sure that I'm on the right track with that. But thank you so much, Craig. We really appreciate it.
All right. Thanks, guys.
Thanks. Yes, I see Drew is here. Thank you very much for joining. Carnivero is a nursery based out of Texas. They have some really amazing things. I have bought from him several times, and I've always been impressed with the wide array, particularly of Nepenthes. Just such beautiful plants that I've gotten, and we're really delighted that he is here to talk to us today a little bit about Carnivero. So Drew, whenever you're ready, just take it away. Thanks.
Okay. Awesome. Yeah. So, yeah, and I am in a bright greenhouse right now. The fans just kicked on. Just let me know if there's any issues. I might be able to move around a little bit. But yeah, I just wanted to give you guys a little bit of a history of Carnivero, as well as what we're trying to do here, and then I wanted to give you a little tour of our facilities around here. Carnivero, let's see. I've been growing carnivorous plants since I was 12 years old. So man, that's been a long time. It's 27 years now. I immediately was really drawn to the Highland plants, obviously, Highland Nepenthes. But the thing is, I grew up in Arizona, particularly in Phoenix. Trying to figure out how to grow them was always a challenge for me. So that actually led me to get into engineering and science. And so that was kind of the route that I went to go. But a lot of that was because I was so fascinated with being able to grow carnivores or just other living things that that required really stringent environments. So Carnivero was kind of a sideline business, throughout college and early professional career, and then let's see, I started building up a breeder collection. Starting about 2008, 2009. That was about the first time I went to Borneo and really saw Nepenthes in their element, just a lot of thoughts came from those first trips. Being like, how amazing these plants are, but also thinking of ways where we might be able to help with these populations in the wild. And what kept on coming back to me is the thought of just being able to sustainably produce these things in a way that would take a lot of pressure off of the poaching that I saw out in the wild. Fast forward, 2016 came along. And we decided to give it a full-time go. And so moved out, left my tech job in the Bay Area, moved out to Austin, Texas, and been running Carnivero full-time ever since. It's really great right now. I've got 10 full-time staff. We're looking to hire 3 more in the next month or 2. So if anybody's interested in coming out to Texas, feel free to send out your resume. We've got, let's see, I'm going to show you guys the greenhouses. We pack them really efficiently. We've got 2 3,000 square foot greenhouses, but we grow on 3 levels. So we use all our under the bench lighting to supplement that, so we get a lot more space out of it. And then I've got an ultra highland grow room here, and then we've also got a really nice lab, and I'll give you guys a quick walkthrough of that as well. And then we've got some other facilities around here too that just kind of help us get all our jobs done on the day-to-day. We're opening up a storefront greenhouse later this year. And that one's actually pretty much completed. So I can give you guys a preview of what that looks like right now. It's kind of it's a little bit messy because we're still putting the finishing touches on a lot of things there. Doing what the fire marshal's asking us to do to get our certificate of occupancy and all that stuff. Let's see. Other thing I wanted to mention is we also do produce our own grow lights here. So like I mentioned, my background is engineering and science. So I actually did my master's thesis in optical engineering on LEDs for horticulture. And so that's kind of where the 4-Wave brand came to being. And a lot of these, the grow lights that we produce are ones that are really specific for being able to grow plants, plants for the collector, plants that can be put in a greenhouse, plants that can be used in a home, not necessarily the high power things that are used in these warehouses where they grow a lot of cannabis. That's where a lot of grow lights are mainly used. A lot of the models that we offer are something a little bit different, that they can grow, you can grow Papillopetalum orchids, or you can grow Aurores, you can go Nepenthes, under these lights, and they're not just going to blow them away. So, I'll give you guys a walkthrough starting now, just kind of what we got going through here, and then I'd love to answer any of your questions. So right now we are in our greenhouse 2 or tropical greenhouse. We do an assortment of climate zones in here just out of necessity. You know, one day I'd like to get a third greenhouse going where it's just temperates, and then we can focus on our bog plants in there mainly, and highlight plants. But for now, we just kind of shade different areas of the greenhouse, and then move things in and out accordingly depending on the seasons. So right now we have, let's see, we're in the highlight area. I'm going to go ahead and flip the screen around. Let's see. So you can see we've got a whole bunch of kind of dormantish Sarracenia flytraps. We've got a lot of picky sundews. That's all Gemme right there. And then we move over here, and we've got a lot of these are bromeliads, other tropicals, bunch of aeroids. There are a lot of aeroids. Philodendrons. Orchids just for a hobby, but we do breed a lot of anthuriums. So, and they just tend to grow really well with carnivorous plants. So it just, you know, we naturally got into them. You know, they grow pretty much the same as Nepenthes. It's just they want a lot of fertilizer. So here we've got variegated bananas. It's the second one that's hit the ceiling. We're gonna chop that one down pretty soon.
Hey, Drew, I think we lost you. We can see your video.
Yeah. Okay, so I think what happened was as I went through the greenhouse, we've got different access points or whatever for internet connection. So I think we're hopping between access points. So I'm going to go ahead. I'm going to go back and you know, show you guys where I left off, and then we'll move over to the other greenhouse. So we're just in the wet wall area. Here is we call this the pingdom. So we do a lot of pinguicula production in-house. We do it tissue culture and through seed. And they really love our 4-wave light. So you can kind of see that's all pings. You got spillover pings over there. And then we just have a whole bunch of Nepenthes propagation right here. See, we've got them up in the rafters. There's young seedlings right there. You can see some of the seedling trays over there.
Hey, we can see you, but we can't hear you.
Okay. Sorry. Again, that access point jumping, I think, is what's happening. But this should be the end of it. I think we're back into a good coverage zone. But anyways, just wanted to introduce you guys to a room that we built. This place, you can see the humidifier going right here. This is essentially a humidity room where we harden off our explants coming out of tissue culture. So you can see we got things. We have Nepenthes. Cephalotus, I mean, anything that's coming out of the lab, it goes in here. And it stays almost 100% humidity for, you know, a few weeks to months, and then we move it on out and get it greenhouse hardened. So, not a really big room, but we do a lot with it. So next up, I'm going to show you guys the main greenhouse. All right, so yeah, so we're gonna enter the main greenhouse. So this is where all the Nepenthes breeders are housed, except the Ultra Highland, and I'll show you guys that at the very end. All right, here we go. So this is the Highland room we're walking into. And so this one is actually air conditioned during parts of the year because it's just so hot in Texas. Right now, the climate stays really nice with evaporative cooling. So you can see we just have a whole Nepenthes growing all over each other here. show you guys some of my favorites in this room. Are some of the giants. You know, right here, this is a Nepenthes Loei cross truncata giant. This is one of the biggest Nepenthes in cultivation, I believe. And you can see it just goes up and up. It's flowering right now. So you've got, this is a Raja cross Veitchii, another really great hybrid out of Borneo Exotics. This has all of the volume and size of the Raja, but with a Veitchii shape. That's really fun. That one, you can see that's a female right there. That one we've been playing around with a lot of pollinations on. You can see right now here the mist is going off. Now our greenhouses all have ultra high pressure misting on it. So it's like the misting that comes out at 1,000 psi, and it creates a really nice kind of instantaneous evaporation effect. So it's not like a wet mist all the time. It's just gets into the air and it cools it down. Right here, we're in the intermediate room, and here's a plant that you won't see anywhere else. This is a variegated Nepenthes Veitchii. And we actually, you grow enough of these from seed, you get to pick some out every once in a while. So you see, like, here's another variegated plant. This is a Song Melancholy cross Bosciana, I believe. And that one, we actually crossed the two variegateds together this year. So we're going to see if we get variegateds out of that. I'm really looking forward to that. A lot of other beautiful hybrids here. So that's an old school Nepenthes Whisper from Sam Estes, Bologna Nursery. Here's one Veitchii table. Got a lot of Veitchii here, harvesting a lot of seeds. Yeah, more Veitchii. And then I'm just going to show you guys the. Well, I'll try to go slow because I don't want to lose Wi-Fi connection here if I can help it. Sometimes there's a switch over here, but you see, like, here's our Lola Nepenthes room, and you enter, and there's a lot of legacy aeroids that we're going to move over to the storefront kind of when the time comes. We've just been letting them grow here, but they're just getting out of control. And there's a lot of large Nepenthes right here. Let's see if there's a decent picture on it. There's a big Northiana. Probably 2 feet across. It's just starting to warm up in Austin, so it doesn't have any great pictures. That's one that this is a room. We've got a great Saracenic collection. Oh, here's some that are, these are the ones we've been fertilizing a little bit heavier. They're really starting to kick off. Nice Zygopegal orchid right here. Okay, so I'm gonna go ahead next. I'm gonna give you guys a quick tour of the Ultra Highland room and explain a little bit about how that was put together, and then I'll show you the lab and then take some questions, I think. So we're walking back this way. All right, so now we're entering the Ultra Highland house. So this room never gets above 75. And so we did this, essentially, we just, we built like a kind of a standard wood room, but then we used closed cell foam insulation to get it really, really insulated. We insulated the foundation as well. So really energy efficient. It uses very little cooling actually, for the temperature range that it goes through. We did build some custom electronics to keep the temperatures in the appropriate range. Here's a nice one. This is Spectabulas cross Edwardsiana. And here's actually I like this clone better. Another Spectabulas Edwardsiana. Now we've got some of the picky Ultra Highlanders. I mean, the motivation for this greenhouse was, a lot of our Highlanders did really well in the other greenhouse I showed you guys, but then there was quite a few that did not do well. Like over the long term, they would do great, like except in the summers, and then in summers they'd always take a bit of a hit, and it really set them back, and then we were just on this cycle continuously. So there's a lot of plants that we weren't getting the flowers frequently, so as we were hoping for, like things like Jambon and Jekylliniae, things like that. Even our Loiais, they just weren't flowering for us enough. So we built this, and as soon as we did, they all took off, and they're just not stopping. So nice Loiai right there. I'm going to go ahead and go to the other side. So undulatifolia right here. Some nice squat pictures on it. Undulatifolia. Zuniae right here. This undulato, I think it looks like Slimer from Ghostbusters. So it's goofy looking. All right, so more Loiai. Look at that. Take a spoon out of that exudate right here. Just moved this Raja in. Had to completely repot it. It had roots. Raja's one that's a very rooty plant. Like, you can see part of the root ball up there. Just kind of from the old pot. This is one that definitely needs a big pot. Needs a very big pot, or else it can run into issues. So whenever we up potted it, we did break some roots. I was a little afraid what it was going to do, but it seems like it's bouncing back pretty well. I'm seeing new growth on it. Over here, we've got some other cool stuff. Here's a nice large Vilosa. Nice red clone. This one is a Edwardsiana Vilosa. It's actually flowering for us. It's a big male, so we're gonna do a lot of crossing with that. This is it's a nice Edwardsiana right here. This is actually a female we got from Jeff Shaper. This is a yellow clone we got from him, and that one again, hoping for some flowers really soon on that one. Yeah, just the Nebular, Rob Cantley, whatever you call it right there. Female coming to bloom. Nice red Edwardsiana. I really like the shape on this one. One of our Loiai truncata in-house crosses right here. This one has a crazy pitcher to leaf ratio. Edwardsiana right here. Look at that. It's not that big of a plant. It's right here, Loiai Edwardsiana. Oh yeah, and Naga. You can't forget that one. This one's our nice. That's a male. We've got a nice female down here as well. These get big and they stay purple. Purple black. And that one's got a bit of powdery mildew on it, but some of the new pictures maybe look a little nicer. Yeah, I can't really get to that one, but it's a really nice plant. We also go Helianthera in here. As you can see. They really enjoy the floor wave lights. More Helianthera on the back rack. These are more of our sales racks here. Nice Hamada. We just moved our Hamadas in here. So now they're really starting to pick up after repot. Been all right. Ooh, didn't see that one. That one, I believe, is it's a Diabolica right there. Finally putting out an upper picture. All right, so I'm going to show you guys the lab next, and then we'll just do a little bit of Q&A, whatever you guys want to talk about. And it is certainly a beautiful afternoon in Texas right now. It's it was cold for a little bit, but now we've been getting 75 and sunny. Pretty thankful for. All the plants are starting to wake up. All right, so coming around to the lab. So coming through here, pretty standard tissue culture lab here. We have our lab kitchen right here where we make a lot of media. Right here's one of the hoods has a bunch of media that just got sterilized through the autoclave right there. We've got our Market Forge autoclave right there. This is one of our workhorses. Yeah, it's general lab kitchen stuff. And then here we've got a lot of, these are the flasks that are on deck right now. These are the ones that we are going to have to be processing soon. So we kind of keep them in this area, or ones that have been recently flasked and we're just monitoring for contamination. So we've got a lot of things going on through here. There's, I don't know, grab some things and show you. Here's some pinks. Things grow really quickly in tissue culture. A little hard to spot the condensation and everything. You guys get the idea. It's pings. We do a lot of steroids still. Here's a variegated philodendron micans. And nepenthes. Oh, we've got nepenthes. And then we do color code all our media, just based on the different things that are in the media. Are there hormones? What's the sugar content? What's the vitamin content? Nutrients, all that sort of stuff. So those are various places. And I'll show you guys the flask grow out room. So this is actually a converted shipping container that we insulated and then just got it going so we could do flasks. And we didn't think it was going to fill up so quickly, but now it's already filled up. So we're thinking about our next lab expansion. But these are there's a lot of plants in here. So these are the ones that are going out soon, getting planted out. Right here, we've got a what is this? This is an Ampullaria intercross that we did. Ampullaria tricolor cross purple striped. A bunch of VGI, a bunch of toothy crosses coming through here. I was pretty stoked that this year we were able to do an F2 Trusmatiensis. So that's a Nepenthe macrophylla across Loia, macrophylla. And those ones actually see termination on those and just starting to take off. Yeah, so that's that's Carnivero in a nutshell. We do have a storefront as well. I'll give you guys a quick show of that, just so you kind of see what the work in progress has been. But you know, it's do understand that it's still not completed yet. But I'm giving you guys a quick preview.
Just a quick question as you're walking. Are we able to visit you?
Not yet. So I'm going to show you right here. We're coming up onto the storefront. So you see, built a nice deck right here, and that's the storefront right there. It's not huge. You know, it's going to be about 900 square feet to begin with, but you know, it should be a nice space. We've got most of it all built out right here, as you can see. We are actually letting our Sarracenia go dormant for the winter in here. Yeah, so we're gonna have a living wall right there. That's gonna be a big living wall that goes up to the top. You know, it's gonna be filled with aeroids and nepenthes. We're just excited to be able to host people in this space. Yeah, so you know, that's our tour. Yeah, happy to take any questions.
That was truly incredible. We have time for maybe like 2 to 3 questions. If anyone has them. There's stuff popping into the chat also about your amazing plants.
Oh, sure. Actually, yeah, I'm not, since I'm holding it right now, I haven't been able to see the chat so much. So if you can recap anything that's on the chat.
Yeah, I can read them out to you.
Sure.
So Jason says, I will be in the Austin area around April 8th. Maybe try to visit your nursery. Kind of already answered that. So seems like stay tuned.
Yeah, stay tuned. We should be open pretty soon. I don't know. It's been a challenge with the county, actually. It's very difficult to build anything in this area of Austin. I wish I would have known that before I bought the property.
Got it. Oral says, I got my Berkei from Carnivero 4 years ago. Still going strong in my kitchen.
Awesome. Great to hear.
Jason says, you need some tree shrews to eat the exudate from those Loei.
Yeah. We get, every once in a while, we'll get a rat in the greenhouse. And the Loei won't get them, but that Loei truncata that I showed you guys, that one is that's a rat eating monster. It gets about one a year.
That's incredible. Helga says incredible. She also says, Drew, thank you for showing us the scope of this operation. It's really amazing. Jason says, your collection and propagation process is really incredible. Blake says, whoa. So fantastic all around.
I have a quick question. Can you guys hear me? I was fighting with my mic here a second ago.
Yeah, I can hear you.
Okay, so I'm looking at your TikTok. I think I wanted to ask, is this your official TikTok? I'm a TikTok influencer, and okay. I have hundreds of people always asking me, where can I buy from where? So I'm always looking for sellers and suppliers. Yeah. And I did look, just a few days ago, I went looking for you on TikTok and I saw, but I wasn't sure if that was you, because there are a lot of imposters and things like that. So
we are on TikTok. We definitely have our account is Carnivero on TikTok. But you know, unfortunately, we just haven't done as much as I would like to, and that's solely just because we just haven't had the bandwidth to really dive into that. But I would love to. I'd love to get more involved there. I know it's a great place for outreach. I really appreciate that. Cool.
All right, thank you.
Yeah, thank you.
We have a question in the chat from Michael. Given the density of your plants, do you have any issues with insect pests? And if so, how do you deal with them?
Yeah, so we have gone almost exclusively to beneficials. It's just because we have, yeah, you're right. We've got a lot of density in the greenhouse. It's something that we're very actively working on. We're trying, you know, we're getting more and more organized as time goes on. You can believe it or not, that is actually more organized than we were a year ago. You know, it keeps on getting more organized, but they keep on growing, right? But let's see. But back to pests though. We've the only pests that we really treat with chemicals anymore are mealybugs. Those ones we tried mealybug destroyers for, and they just don't cut it. Unfortunately, they just don't breed quite prolifically enough to really take care of the problem. But everything else, our go-to is beneficials. We're talking scale, we're talking thrips, we're talking mites, any of those. Like there's a predatory insect or organism that will just take care of them. And then the great thing is you don't have to worry about any pesticide resistance. And also the thing is we've got 11 people working here on any given day. It really sucks to have to shut down a greenhouse for a whole day. Just because we're spraying something that might be, like have a long return entry interval. So the beneficials are great for that too, because they're just bugs. They're just bugs there. They're nematodes. They're predatory mites. They're predatory wasps, something like that. And it's just a continual ecosystem that we have growing or have thriving in the greenhouse. So I'm happy to, and if anybody's interested in the predatory insects, especially you at home growers, because I know a lot of people don't like to use chemicals in their homes. I'd be happy to refer you to the insects that we use.
Michael says thanks. Craig says fantastic lab set. Wow, that is a lot of plants in plastic containers. Great job. And then Jason says, I am super impressed by how you handle the insect pests. That is really great to hear. Thank you. I think for purposes of time, we will cut it there. But I just wanted to say thank you very much. And we look forward to seeing more plants from you.
Okay, sounds great. Thank you for having us. And if you have any more questions, just shoot them over on email, service@carnivero.com.
Awesome, thanks.
Yeah. All right. Well, and thank you for hosting this as well. This is just great seeing other growers.
We're so happy you could join us. Thanks.
All right, thank you. Bye.
All right, so at this point, I would like to introduce Jason, who is going to be our leader for the summer field trip to the Pine Barrens. Jason was actually a steering committee of MACPS a while ago, did some incredible work, and we're so happy to have him come and lead this field trip, which we really have been talking about since essentially the beginning of this society. So Jason, whenever you're ready, just take it away.
Hey, thanks a lot. I appreciate it. I think, am I sharing the screen correctly now? You can see sphagnum moss.
Yes.
Okay, great. Then I figured out how to do something right. I am actually really shocked that you guys haven't made it down to the Pine Barrens yet. I'm honored that I'll be your first intro to it. It's pretty cool. This is I'll try to make this very, very brief so we can move on to the rest of the meeting. But this is just going to be a very brief overview of some of the plants that we can hopefully see. And maybe you guys will do better with the Utricularia than I will do with the Utricularia. But here's a few possible sites that we may wind up visiting. We'll stop by, actually our meeting point, rendezvous point in the morning should be my bog garden in Toms River, New Jersey. It's a 16 by 16 foot bog garden filled with carnivorous plants and a whole lot of non-carnivorous companion plants. It's all temperate growing, except that I so far have had Drosera binata in all of its forms for about 4 years and Drosera capensis for 2 years, but just from the Guaido Pass. That's the only one, and it sort of gets through the winter. And then we'll meet there with my co-host, Jason Austin, who some of you may well know. He used to work at Rare Fine Nursery, and we grew carnivorous plants there together. He'll be joining us to host for the rest of the tour of the Pine Barrens. One of the most likely the first stop that we'll hit is going to be a famous site called Webb's Mill Bog. It's right off of a very accessible highway. And there is a very nice boardwalk that goes through the entire bog, which allows us to access everything and see all the plants without getting muddy and dirty, but more importantly, without damaging the bog. Some of the plants that we'll definitely be seeing there are Sarracenia purpurea, Calopogon tuberosus, and Pogonia ophioglossaides. The grass pink orchid and the dragon's mouth orchid will most likely be blooming at that point. There's a small patch of curly grass fern, Schizia pusilla, a pretty large patch of Narthecium americanum, which may not quite be blooming at that point, but it might be. And there's quite a few other things to see there, and a beautiful fen habitat. I will be checking that site out ahead of time just because there have been, for the last number of years, family of beaver that have been flooding the site regularly. If it's flooded too badly, then I'll probably take us to a different site because it won't be worth seeing because of the flooding damage, unfortunately. But we may get lucky. Someone has been removing the beaver dam occasionally. And we'll talk more about that when and if we do go to that site, but most likely we will. Next stop, and this one will be pending. I'll be discussing this with Kevin a little bit more in future chats. But it's the Franklin Park Preserve. It's a very large preserve. I don't know the acreage on it, but it's really quite significant in Burlington County, New Jersey. It used to be the site of a family-owned cranberry bog or a complex of cranberry bogs and blueberry fields. They've been converted by the state of New Jersey and the New Jersey Conservation Foundation. And we may be able to host our lunch and have the meeting and auction there as well. They've got fairly good facilities there for us to potentially use. Then from there, we'll go on a fairly short run south to the old town of Martha. It's an extinct town in the middle of Wharton State Forest. We'll go down there to an old ironworks site that has a really intact bog that's relatively easy to access. I'd recommend if you have four-wheel or all-wheel drive that we use that, and if we could jump in as few cars as possible, that would be helpful. We'll figure out how to make that happen. That's the sites. Well, hold on one second. Yeah, there it is. Some of the things to bring is Kevin had mentioned to me that we are going to be going rain or shine. So please dress appropriately for the weather to keep yourself comfortable. We'll be getting wet and muddy, at least at the ironworks site. So bring again, everything is about being appropriate for the times. People are going to wear muck boots. They're not my preference because there tends to be a lot of destruction when muck boots are involved. You're insulating yourself from the habitat, and it's really easy to just sort of step on all of the rare and valuable plants that we're there to see. I go barefoot into the bogs. That may or may not work for you guys. I'd recommend it. It feels really great in the sphagnum moss. But any kinds of decent strap-on sandals are really great, or sneakers are very good as well. Bring a change of clothes and footwear if you can. It'll just make your ride home. I guess some of you might be coming from quite afar. So it'll make your drive home a lot more comfortable and help you keep your car cleaner. I don't wear repellents. I don't really have any problems with that in the Pine Barrens, but I will carry stuff with me in certain times of year just in case we encounter ticks, but that won't be a problem this time of year, or at least not the seed ticks. Bring plenty of water. You can leave it in the car. We're not going to be far away from the car no matter where we stop, but bring plenty of water. And Kevin and I will be working out the lunch details and where we're going to be going for lunch, whether it's the park preserve or even just a local Wawa, something like that. So we'll keep you all posted on that. If you guys do photography, bring your camera gear along. And lastly, bring a very profoundly strong sense of caring and caution as we enter these really fragile habitats. I think some of us are used to seeing wetlands that recover quickly from man-made destruction like footprints. These are very, very eutrophic habitats. They don't have a lot of nutrients and they recover very, very slowly from even just our footprints. So going in with a lot of care is going to be very critical for us so that we can continue to enjoy this site in the future. And next is just a quick tour of some of the plants. This is something that you probably all recognize, Drosera filiformis. We'll certainly see this at Webb's Mill and at the ironworks site. It's kind of common in the Pine Barrens. I believe that the Pine Barrens is the strongest bastion for this species in its entire range. I've heard anecdotes from the New England populations that they're not nearly as robust as New Jersey. It's extirpated from Delaware, and it's only at one or two small sites left in North Carolina. And then the Florida stuff is a different animal altogether, as we're probably all aware. Let me do myself a quick favor and get rid of this. There we go. Now I can read the whole slide. Drosera intermedia is the other common sundew that we'll see, one of the other two remaining common species. Again, we all are familiar with that one. That'll be at Webb's Mill and at the Ironworks site. Third species in New Jersey is Drosera rotundifolia. Again, one of the probably the most common sundews in the entire world with the widest range. And we'll see that throughout anywhere we go where there's moisture. A quick overview of the three of the species again, just up close of the leaves and the flowers. This is an interesting shot. I actually have to confess I cheated on this shot. So the Drosera hybrida, the natural hybrid between Drosera filiformis and Drosera intermedia, occurred at this spot naturally where it's growing. I planted a Drosera bellesiana from a cutting that I took from a plant that was about 50 feet away, right next to the Drosera hybrida, just so that a few years later I could get this shot. So this is both sundew hybrids that we might encounter in the field. Drosera bellesiana, which I guess most people are now calling Drosera eloisiana. I will not get into that debate here. But that one is actually really common in the Pine Barrens. There's a good chance that we'll find that as long as we get into some of the decent sphagnum buds. Drosera hybrida is very rare. This is some of the diversity of pitchers that we might expect to see. These are all New Jersey pitcher plants, except the dark red one in the bottom right is one from Massachusetts called Merlot. But the rest of these are New Jersey typical, and there's quite a bit more variation than this even. For those of you that don't know, Sarracenia purpurea in the Pine Barrens is an integrated population between Sarracenia purpurea from the north and Sarracenia purpurea venosa from the south. And so in the Pine Barrens, we get tons of variation, anywhere from long, skinny, glabrous plants like pure purps, all the way to plants that look just like Sarracenia rosea from the Florida Panhandle, and everywhere in between. So it's pretty fun and interesting to go trotting through the bogs there. Next, we move into the Utricularia that we might be able to see. Some of them will probably be blooming when we're out there. Others bloom later in the season. Sometimes they're completely dependent on moisture levels and temperatures. So July and August is a good time to see some of them, but some we should be able to see. This is Utricularia cornuta, horned bladderwort. It's just one of, I think, 17. I think it says however many there are in New Jersey. So cornuta, gymnoscopa, Gibba inflata, intermedia, Juntia, macrorhiza, minor, olivaceae, purpurea, radiata, resupinata, striata, and subulata are all native to New Jersey. Some of them we will definitely encounter. We'll just have to see which ones. This is, let me move this again so I can read it to make sure. Yes, it's the Utricularia gibba that I found a few years ago. Apparently, I think if I recall, that's the most widespread Utricularia in the world. Utricularia inflata. I know a few sites where I can encounter this. Not likely to be any of the spots that we're going to go to, but I can try and see if I can locate any for us. Utricularia macrorhiza. This one I don't expect to encounter in the Pine Barrens. I see this up North Jersey. As far as I know, it's not really known to grow in the pinelands. So that one was just included because it was one of the Utricularia of New Jersey. This one we might see, and it might be blooming. Utricularia purpurea grows in open water in the Pine Barrens, and it blooms in these large masses. Typically, it's quite fun and impressive to find. The next one is Utricularia resupinata. This is not one that we'll likely see. It's very rare to find it in bloom. I found a super bloom a few years ago. I was very lucky to find that. I found it actually in 2 locations in New Jersey, which was pretty surprising. But you never know. It may pop up. It does weird things, and it shows up in places where it hadn't been seen before. So we may get lucky. Certainly we'll be there at the right time of year, I think. Utricularia striata. This patch is actually one that has been blooming for years and years and years, pretty reliably at the backside of Webb's Mill. We can see this exact view from the boardwalk where we'll be entering the bog. So hopefully we'll get lucky. It will be the right time of the year as long as the water conditions are right. And this down at the bottom is the foliage, the traps and leaves and stems of the Ustriata. This is the red foliaged form of it, which I was told by some Utric aficionados is quite rare, and they were very interested in it. Utricularia subulata. We all know subulata because we all have it in all of our collections ubiquitously everywhere around the world. But it also grows wild in New Jersey, and we will probably see it popping up out of the sandy sphagnum edges along the bogs. Another interesting plant that's only newly recognized as carnivorous, and this one not exactly, but you probably all recall in 2021 the big media blitz that Triantha occidentalis was labeled as being carnivorous, catching small insects on the sticky hairs along its stem. Well, we have a unique version of that. We have a species in the pine barrens of New Jersey that only occurs in Burlington County, New Jersey, and nowhere else in the world. Its stems are also very sticky, and I have photographs of my plants from my bog garden showing it capturing small insects. Now, in order to claim it actually carnivorous, the proper research and tests would have to be done so that we can see that it's absorbing the nutrients from the insects and using that nutrients to grow new plant tissue, as was done with Triantha occidentalis. But at least we've got the first step in the process that's demonstrated here, that the glands there are sticky and produce this exudate which covers the whole stem, and you can see the stem later in the season. There's tiny remnants of little mummified digested insects, or possibly digested insects, all along the stem. So that's a quick overview of the carnivorous stuff. And here's one of the orchids that we may get lucky to see at Webb's Mill. That's been declining in recent years, but the Arethusa bulbosa, the dragon's mouth orchid, does bloom there, and we may be able to get a view of it, especially if you have binoculars. It's easier to see them from the boardwalk with binoculars. A second one is the snake mouth orchid, Pogonia ophioglossoides. This used to be in very large numbers at Webb's Mill, as is the next species that we'll see. The beaver flooding has damaged these populations pretty significantly, but we should be able to expect to see this one and the grass pink orchid Calopogon tuberosus blooming at the bog at that time. Additionally, we may stop and see this on the way down to the ironworks. It's a rare milkweed. It's rare in New Jersey anyway. Red milkweed, Asclepias rubra. There's a little roadside ditch that we can just pull over and look, basically out the window of the car if we want to, and just say, oh, yep, there it is. And, but for us in New Jersey, it's an exciting plant because it's really quite rare, and we're at the northern extent of its range. Narthecium americanum is a listed species. It used to be found in Delaware as well, and I believe there was records in North Carolina. It's extirpated from both, so now it only occurs in the New Jersey pine barrens. This shot is from Webb's Mill, so hopefully they'll be in bloom and we'll be able to get a look at them. It's quite a large population of them there. Some of the polygolas that we'll probably see just kind of creeping out among the bog plants are Polygola cruciata, Polygola lutea, and Polygola sanguinea. These three are just nice little denizens of the bogs and the sphagnum tussocks and wet roadside ditches. It's always pleasant to see, and it's easy to spot the orange ones from the roadsides when you're driving by at 60 miles an hour, and it's a good tip off that there's probably some other really cool stuff in that roadside. Some of the club mosses that we'll probably come across are Lycopodiella alopecuroides, Lycopodiella oppressa, and Pseudolycopodella caroliniana, which is great fun to say at parties when you've had a few drinks. These guys are all pretty common in the pine barrens, except that caroliniana, which I believe is an S3. It's listed as vulnerable in the state. And something that we'll see probably at both bog sites that we stop is the Ericolon aquaticum, seven-angled pipewort, which is pretty common, and we should be able to see that sticking itself out of the water like this. This is the curly grass fern. There's one small patch of it left at Webb's Mill that, again, if it's not covered in beaver flood water, we'll be able to see it. It's a small rare fern that was actually described from Wharton Forest in the pine barrens. So it's unique, special New Jersey heritage. plant, and it looks like green pubic hair, so everybody kind of gets a kick out of that. Um, and this is just one picture of a beautiful fen. I was hoping that we could go and visit this one, but it's a bit hard to get to. And when you're there, you're generally going up to about your chin in muck. So I figured it was probably not the best stop for the whole group, but it is a pretty spectacular site. So maybe one of these days, if you all come down in small groups, we could carefully go out and check it out on our own. And then if there's any questions or comments or suggestions, I'm happy to field that. I think I'm going to stop the share now and leave that to Kevin to interact with the rest of you folks and go with some questions or comments if you have any.
Awesome. Thanks so much, Jason. We are super excited to go on this trip. Any questions?
I'm glad that you're enjoying the photos. I enjoy taking them, so I appreciate that. And like I said, Kevin and I are gonna, rub our brains together a little bit in the near future to decide on specifics with lunch, whether we're gonna need to make, orders ahead of time if it's a pizza joint or things like that. So he'll communicate to everybody, and we'll get it all sorted out.
I have a question.
Yes.
So you said that you usually go barefoot. So I guess after the Black Shannon trip, I like spent some time in that bog barefoot in shorts, and my skin got like sliced up and was like so irritated that I was like, do I need to cut my camping trip like short? And I like have pretty sensitive skin. So I was just wondering if that's like a thing that has ever happened to you, because I don't know if it was like a particular like plant that irritated my skin, or if it just kind of the grass maybe like.
It's probably the grass. I've
Yeah.
been to Black Moshannon, and there's a lot of sawgrass there, and
Oh,
sawgrass
okay. That
shreds you. Yeah, I
okay.
hate sawgrass. Fortunately,
Yeah,
it's not
sawgrass
really
sounds like it would do that.
yeah, probably it's not really an issue in the Pine Barrens. It's not one that I've
Okay.
run into. Um, the only thing that really ever is a problem for me going barefoot in the Pine Barrens is um, after a burn, a lot of the cedar trees will burn down to a nub at the base near where the trunk is. And then that nub will decay just enough so that the heartwood sticks out like a nail, and that will puncture your foot occasionally and leaves a big black mark in the puncture hole where the char basically tattoos you temporarily. Um, that's not really too much of a problem walking on land or in low bogs, where you have to walk carefully and you're being agile about how you place your weight because you're being sensitive to your feet. That's more of a problem that I run into when I'm in deep bogs where you're going and then you hit an unexpected deep spot that you had no idea was there and you plunge in. And then when your plunge is stopped, it's usually stopped in one of those sunken cedar trees that's burned maybe hundreds or thousands of years ago. That's a long, long explanation that to explain. I don't really have very much in the way of problems in the Pine Barrens. Um, if anybody does have concerns, wear long pants and wear sandals or flip-flops, or sneakers are another really good alternative. They're still light. You tend to do a lot less damage going through a bog in sneakers than you do in boots. And long pants will solve any of the problems with, Smilax, vines that are reaching out at you or any of that kind of stuff.
I do want to quickly say, we have multiple sites planned on this trip. Some, like Webb's Mill, will be a boardwalk. Some will just be going to visit Jason's bog. And so, whatever you are comfortable with, feel free to come along for those. If a place is, too out there for you, do what's comfortable for you. We're not going to pressure you in any way. Feel free to join whatever portions you would like.
Yeah, no, this is all for fun, and I am sure that whoever's able to join us on the trips will have a blast. So, like I said, it's all about being comfortable. You have to know your own limits and what you're comfortable with, how wet you want to get, and whether or not you're prepared to change your clothes so you can just sort of throw caution to the wind and totally muck yourself up if you want to. There'll be plenty of opportunity for that. There's also a river right next to the ironworks site, so you can wash off in the river. So you're not going to be like completely mucky. You'll just be wet and a little bit dirty by the end of it, because you can wash off easily. One of the other sites that I was hoping that we could get to, I took Jerry out there years ago. It was a really fantastic, large fen complex, and we removed a bunch of non-native Sarracenia from that site. And it's a beautiful site to begin with, but it also has a really nice little swimming hole adjacent to it that's good for washing off in. But fortunately for the plants and the habitat, unfortunately for us, that site has had the access closed off to it in an effort to keep out the off-road vehicles, which have been doing unimaginable damage to the environment there for a few decades now. And so NJCF, the New Jersey Conservation Foundation, that now owns the land, is trying to keep out those vehicles, which includes our vehicles. So we can no longer access that site, unfortunately, unless we wanted to hike in, which would not be absolutely impossible, but it would consume the rest of the day. It would be a one site visit day. And I figured it's more important for you guys to see multiple sites as much as possible. And let's see. There are many signs of, really more signs of poaching at Webb's Mill. Oh, we'll discuss that when it's not an online forum that has a time limit. Yeah, the poaching, great. Well, hopefully there's still plenty of pitcher plants to see there. I did notice some losses last time I was there as well. Oh well, sad to hear that. But yeah, anyway, that's pretty much my rundown. Hopefully it wasn't too far over the time limit.
No, that was perfect. I think if
Good.
people have other questions, they can feel free to email us, the regular MACPS email, info@macpsociety.com, and I can route them appropriately.
Yeah, sounds great.
Yeah, we look forward to seeing, hopefully, a lot of you at the June 20th.
More than my head and shoulders, at least.
Yes, and so we'll send out more information as the time goes by with more details.
Great. Excellent. Thank you.
Thanks. At this point, I would like to hand it over to Hannah to run our photo contest. As this is playing, just wanted to say thank you to everyone who submitted the photos. We truly have some amazing submissions this year. Notice there are a couple of slides that say cast your vote. So the MACPS board went through and actually chose 5 top photos. So please help us choose the 2 winners by voting at the very end. So at this time, you should see pop up on your screen all 5 of those photos. Please go ahead and cast your vote. Awesome. Thanks everyone for voting. So it looks like number 19 was the winner. Nepenthes truncata crossed with Edwardsiana, predatory plants with mantis. And then the second place winner was Drosophilum lusitanicum. So we will be notifying the winners of those shortly. You'll get an email from us. Thanks everyone for participating. With that, I would actually like to hand it over to Rick, who will be telling you a little bit about MACPS Connect, a new initiative for members.
Yeah, thanks, Kevin. I appreciate that. The photos are fantastic this year, by the way. I really appreciate everybody submitting for all of those. They really turned out awesome, and really happy to see number 19 actually win. I thought that was my favorite as well. It's a really, really stunning compilation there of everything that's in that photo. So cool for sure. So I just want to go ahead and take a moment to kind of briefly introduce the new initiative that we're going to be doing. It's a monthly meeting of sorts that I'm going to be starting off. Basically, the idea behind these new monthly meetings is going to be focused a lot more on engagement with members directly. So we want to get everybody more involved. And obviously, it's kind of hard to do that in a physical capacity as often because we are as spread out as we are. So to bridge the gap, what I'm planning to do is come up with multiple types of sessions every month where we can all get together and focus on a specific topic that is, obviously centered around carnivorous plants in some way, shape, or fashion. So they're going to be varied in nature. You may find that there are some things that you are really fascinated in participating in, or other things that, might not exactly be right up your alley, but, there's going to be something new every month that you can hopefully find, that would be interesting to you. So right now, this is just going to be a preliminary introduction of what those meetings are going to be, and we've rolled out concepts for what the first 3 meetings are going to be focused around, but definitely be looking out for more details surrounding each one of those as we kind of roll into the following months. The plan for them is going to be held every month, roughly on the 3rd Saturday of the month. That could have some variation depending on who else might be involved in those meetings or what else we might be doing in a given month. So there may be some flexibility there, but we're definitely going to be very prominent in communication about those meetings going forward, when they're at, what they're going to be about, what are the details, all that good stuff. So just give everybody a heads up. For the first 3 months, starting off in March, the plan is to have a cultivation spotlight. It's going to be centric around butterworts, and both tropicals and temperates. We're going to be talking about tricks and tips to cultivation with them, on both sides of it, both how to grow them temperately in different climates, and how to what are the best ways to achieve success with tropical varieties as well, and dealing with those middle dormancies that can be experienced with those. From there, the next month we're planning to do a sort of a show and tell. So members are going to have the opportunity to share and talk about extensively their favorite plants that they're growing. It's going to be an open discussion in that capacity, where everybody's going to be sort of just sharing what their favorite thing is, why it's their favorite, really just a chance to showcase that plant that you're extremely proud of growing. Hopefully by mid-April, it'll give everybody a chance to be able to participate in that, whether or not you're growing mainly temperate stuff or tropical stuff. You'll have some good stuff that's hopefully starting to look nice at that point. So hopefully that'll be a good opportunity to participate in that at that point. In May, we're looking to roll out a sort of new template for what would be a plant swap, where we're going to try reintroducing the open platform to be able to trade plant material between members, and be able to kind of openly share and exchange with that, so that if you've got a plant that you're looking to maybe find a new home for, or you're just interested in seeing what anybody else is looking to put out there, or say you got too many Saraceni, you want to swap it out for some pings, give that a try. We're going to be making that open and available, so everybody has a space to do that and openly talk about it. On top of that, we're also looking to introduce a sort of virtual auction platform that will go alongside that. So we're still working out the details of the who, the how, and all that good stuff. But we definitely want to introduce it as an element. So definitely be looking for the details on that too. But we're definitely looking to do more auctions that both members and other collaborators can get involved with in time. But that's the gist of it from there. I do also want to put out you'll probably see a posting here in the very near future, on top of what we've already suggested as a sort of plan to go forward. I'd love to hear from everyone on any ideas or programs in particular that the members would love to see talked about in detail. Again, the whole point of this is to be a lot more engaging and collaborative. So if there's a specific topic or type of engagement that you'd like to see, we'll be posting a social post about that, and I'd definitely love to hear from everybody on that in terms of what they'd love to see from us, because I'm definitely looking to meet that as best I can. So definitely look out for more details. Like I said, the butterwort one will be coming up first. So we'll be highlighting that very soon in the near future. And definitely, if you are a grower out there who's trying one or the other, and you're having mixed success, or even if you're doing really well, we'll be having some experts to sort of guide and talk on that subject. But with that said, that's all I've got for it for now. So definitely stay tuned, and I think it's going to be really exciting. Thanks.
Awesome. Thanks, Rick. So at this point, we're going to just kind of open it up to everybody. I thought it'd be nice, just in the spirit that this is a virtual meeting, we don't get to see everybody. If you wanted to, no pressure, but if you wanted to turn your cameras on, maybe we can go around, say, who we are, what got us interested in carnivorous plants. And then if you have a favorite plant, if you want to just talk about that and show it off for us, that'd be great. So just, I can start it off. I've been growing carnivorous plants since 2003. So at this point, it's been over 20 years, which is kind of crazy to think about. I've been part of the society since the beginning, and I will show you my favorite plant, which hopefully you can see. This is actually Nepenthes veitchii, crossed with, Nepenthes mirabilis crossed with veitchii. So you can see the beautiful peristomes on this plant. Quite nice pictures. I've had this, not sure, I think I've had it for 2 years now at this point, but it's grown very, very fast. So that's one of my favorite plants. Encourage all of you to talk a little bit, just you can go randomly however you like, show off your plant, whatever, and we'll just open it up for discussion.
That's a gorgeous plant, Kevin. Where's that from?
Honestly, I got it off of some kind of Facebook group. I think it was just someone was selling these beautiful seedlings. I thought that's a beautiful plant, and I got it. And it's definitely been growing very well for me, and it is truly beautiful.
Yeah, that's stunning. It's kind of reminiscent of what's that popular cross called? It's something melancholy.
Melancholy?
Yeah, that one reminds me of that a little bit. Very vibrant colors and candy striping on that.
Yes, agreed. Anyone want to be brave? Go next. We're a friendly group of people. We can't bite through the screen.
So I'm Leslie Morris Smith, and I'm chairing an exhibit of indoor and outdoor plants at the Flower Show, booth 66. And it's mostly gesneriads and African violets. And then it's the Fern and Wildflower Society. So you guys are my favorite wildflower. And so I invited Kevin to see who else might want to drop off some plants from your society onto my shelves. And then I can give you tickets to the flower show for that day. Or if you're interested in coming many days, just let me know. And I can put a sign up link in the chat. But this is what I look like if you're talking to me through email or text, right, Kevin? So anyway, thank you for, anyway, I've been a member for a couple of years, ever since the first COVID year at FDR Park, when we highlighted Sarracenia and related bog plants. I've been fascinated by them, and I think I made it through pretty good this winter. So anyway, nice to meet you. And if you have any interest, don't let cost be your barrier. Just contact me in the flower show. And that runs March 2nd to March 10th. So it's this coming Saturday to the following Sunday. And Kevin, you and I have to talk about how you're going to take your plants out. And but we have storage. He's going to be able to store the cartons for his bell jar in our basement store, secure basement storage. And anyway, it's pretty interesting, I think. And it's not everybody's cup of tea, but hello, let me turn it over to the rest of you. And hi, Jason, by the way.
Yeah, Leslie,
It's good to see you.
Just want to quickly say we did send an email about this, but if you are still interested in volunteering, please just send us an email and we can get you hooked up for the Philadelphia Flower Show. That is.
It was good to see you too, Leslie. I'm glad to see that you're still involved with everything.
Yeah, very much so. I keep an eye peeled out for your presence anytime, and that field trip days is a killer plan.
Well, yeah, I hope it works out. I think it'll be a good one. Thanks.
I also, by the way, I'm field trip chair for the Fern and Wildflower Society, with the previous one stepping down. And so
Okay.
I'm really eyeing, I want to talk to you guys about possibly sharing that with my group, but I know it's a member only thing, or, it could be a member only thing, but Looks great.
Well, members of the Fern Society could certainly become members of the MACPS.
True. And you're one of them.
I'm actually not, surprisingly. I dropped off the radar years ago. I have not been involved for a long time, but I definitely need to reconsider that.
Yeah, it's a 3-year membership, so I think you only dropped off this year, this December. So.
Yeah.
All right.
I wanted to thank everybody for hosting me and thank Kevin for getting me on board here too. I appreciate that. And I really look forward to showing people some of the places in the Pine Barrens that are so special, especially Webb's Mill. If it's in good shape without any beaver damage, I think everybody will just be madly in love with that place. And it's an easy visit for anybody going through. The most common stop is people that are going to the Jersey Shore. They just pull right over. It's a 100-foot walk to the boardwalk, and it's pretty fantastic.
Hey, shout out to John Hollister. He's the other one who volunteered to plant in the flower show. Thanks, John. So I'd love to hear your voice, but yeah, there you go.
I'm looking forward to it.
Good.
Yeah.
Thank you.
By the way, I mean, given I gather about the Pine Barrens trip, is there any possibility of seeing Lygodium or finding a Lygodium locale?
No, not on that trip. Lygodium is very rare in New Jersey. And I personally only know of one site. I'm sure there are others, but I only know of one site, and it's quite off the beaten path from any trip that we would, any forays that we would be making. But it's a great ask. It's a fantastic plant. I just went and photographed it a few weeks ago.
I'll go ahead and show off this guy here. This is one of my favorites that I've got growing at the moment. It's Borneo Exotics' new Velosa by Hamada. One of their, it's a micro prop clone, so there's a little bit of variation. So there's not just like one, genetic individual, but this one I got pretty lucky with. It's got a really nice coloration to it. When the peristome first opens up here, it's like a brilliant kind of like golden yellow color, right around here, and then it slowly darkens up and becomes this like lovely, red color here. That the body's pretty much dark immediately. You can see, here's one that's coming up, so it's already got like awesome color right off the bat. But when this peristome pops open first, it's just like this two tones, really dark, really light, gold and red bicoloration, and it's growing super fast, compared to either species, Velosa or Hamada, which both grow super slowly. I just repotted this the other day, and it was like, this big last year. So it's definitely humming along quite well, and I'm really, really pleased with it. It's been an awesome plant to grow for sure.
That's gorgeous. I love seeing all of your ultra rare highland plants all the time.
Hi, Kevin. Hi, group.
Hey, David.
I don't really have a plant per se. You know me as kind of a ping guy, a Mexican ping guy, and I definitely have some Mexican pings I like. They kind of keep my mood up during the winter. But a majority of what I really like are outside, and I'm looking forward to trying to get into the Pine Barrens. I can't tell you how many mornings I'll sit out in the bog, well, outside the plants as the sun's coming up, or maybe not that early, but and seeing the dewy filiformis and some of those guys with the first sun on them. So you can see kind of a picture up behind me of some of them. The temperate carnivorous plants are just so beautiful, and the tropical ones get me through the winter. But you guys are so much more better at the Nepenthes and all the crazy things. The thing that's really got me going for the year is I did purchase over the winter a small greenhouse. So in the next couple of weeks, I'm putting up a small greenhouse to try to extend the outdoor season a little bit and hopefully provide a little bit more comfort for some of the plants. It won't be heated, at least for now, but I'm really looking forward to it. It's kind of been a little bit of a purchasing project over the last couple of weeks, but I'm hoping by the end of March it's definitely up and got plants in it. So be looking forward to that.
David had some incredible plants at the recent event that we were at in upstate New York. Some truly beautiful butterworts.
Yeah, I'm looking forward to get my hands on some of those next time we meet up, David, for sure. There's definitely a couple species you got that I'd love to get my hands on for sure.
Well, I made some pullings for you off of that one.
Well, thank you.
And if I could, we might be heading back to New York towards the end of the summer for the fall program, but we're still kind of hashing details on that one. It'll be a much larger arena. It was kind of exciting a week or two ago. Almost 2,000 people came, and they were turning people away because it was so crowded, and now they're going to move into a indoor arena in Syracuse where they'll have upwards of 30,000 square feet of display area and can handle crowds in the thousands much more better than they did the last time.
Anybody have any I know Drew's video was cutting out quite a bit there. But did anybody have any particular favorites that stood out during that whole thing? I know I certainly did, but anybody else have anything that stuck out to them there?
I don't have an answer to that, but and Blake, everyone. I was gonna show off the Nepenthes poi dog that I got from you a while ago, Rick.
Yeah.
But my like actual animal dog is sitting on my lap, so I like can't get up to get it. I'm still having a hard time with like I guess the humidity. I don't know, it doesn't put out a lot of pictures, but it is getting a lot bigger and it like takes up a big space. And I just have it like in my windowsill. And yeah, it's gonna turn into quite the massive plant, which makes me happy because this is the only Nepenthes that I have. I do not do well with them. So yeah.
Well, I'm just so happy to hear that that it's doing all right for you and that it's getting really big. I mean, that's awesome for sure.
Yeah, but I like haven't killed it.
Yeah, I was thinking. I mean, it was a pretty indestructible cross I got from Leilani. So I was thinking like it's definitely something that I think anybody would do well with for sure. So that's great to hear.
Yeah. I am a Nepenthes grower.
It's official.
One single Nepenthes. I think because I really like, I mean, right now, I used to have more outdoor space, so I had some like Sarracenia, but now it's like I pretty much just have windowsill, like stuff that I can grow in my windowsill, and then I have like a tiny terrarium, so I don't have like a bunch of grow lights or anything.
I'll introduce myself. I'm Samara. I'm one of the board members. All my plants are in the other room, so I can't get them to show right now. But my favorite genus, I guess, is the Nepenthes. I can't think of a favorite right now. Just wanted to thank everyone for coming to the meeting and welcome.
No, I'm definitely envious of the Nepenthes folks. I admire them. They are, I don't want to say kind of a grotesque plant, but they are definitely on the farther side of, unusual. But yeah, I don't do well with Nepenthes, and I would like to throw out with to Jason for the New Jersey trip, the Utricularia. I mean, I just between the winter with the Mexican pings and then in season with some Utricularia and some Proceras and Sarracenia, I just feel like I'm bubbling. I can't wait 4 more weeks until things start coming up and getting out and uncovering them. I just can't wait.
just want to say hi. Not feeling so great, so I'm not going to be getting up to show a whole lot of stuff, but I want to thank you guys for being such a great group of enablers. Started in Carnivorous Plants around 2018 or 2019 when Sheila brought me to one of your meetings. And my name's Jacqueline. If you look at my profile picture that's up on the screen right now, that's a screenshot of the greenhouse that is the result of having gone to that meeting. That none of that existed before this, but yeah, carnivorous plants are a bit of a slippery slope, and I will say Nepenthes are my favorite, so that's most of what's in that greenhouse. But there's also now a pretty good collection of pings, and in the summer there are bogs with sarracenias and fly traps and sundews and companion plants. So thank you guys for being such a great group of enablers.
That is so awesome to hear. I mean, I see pictures of your setup, and I'm just truly blown away because honestly, you have some incredible plants, and I'm super happy. Thanks for that comment.
I guess I'll say hi, everyone. My name's Casey Miller. I'm also one of the directors on the board. I grow mainly Sarracenia and Venus flytraps, so they're all in dormancy. I keep them in a big walk-in refrigerator and plastic totes, so I can't share any with you right now. But if you see my picture behind me, that's in June of a couple years ago when they're all flowering and looking great. So I hear you, David. It'll be a few months before my plants are ready for show time, but looking forward to it. And thanks for coming, everyone.
What species is that in your photo?
That particular one is a Leucophylla rubra hybrid that just makes tons and tons and tons of flowers, and they smell really great too. They smell like violets, which is kind of cool.
Yeah, that's beautiful.
Hi, my name is Tim. I am from Maryland, and I'm new to the society, and this is my first meeting. And I'm glad to be following behind Casey because I was feeling as though I'm the only Venus flytrap keeper in the group, and but I haven't been doing this very long. Had Venus flytraps when I was a boy, but back in the time when you bought them from the back of comic books and so on. And I'm looking forward to learning more. I do have a couple pitcher plants, but I have predominantly all Venus flytraps. So thank you. I've really enjoyed this meeting very much.
Welcome, Tim. I think you'll find definitely we have our own preferences, but I think all of us, well, I'll speak for myself, I guess. I lean towards Nepenthes, but I have a very sizable Venus flytrap collection and an even more sizable Sarracenia collection. But as others have pointed out, those look terrible right now because they're all under 8 inches of pine needles. But I do enjoy collecting Venus flytrap cultivars, and I'm definitely one to see that's interesting. I'll buy it for $80 or something like that. So I'm with you on that.
We should say for those of you who are new and haven't been to one of our in-person meetings, they're really great. They're not always field trips. We have different in-person meetings over the year, and they always feature a auction where you can get your hands on lots of different types of plants grown by members. So if you're into different types of plants, whether it's pitcher plants or fly traps or Nepenthes, definitely check out our in-person meetings.
I'm kind of like Casey, like Blake, where I have a limited amount of area that I can use, and I'm keeping them indoors in aquariums. I would love to go outside, but my wife keeps bees. So if I had the carnivorous plants outside, they'd probably be eating all the bees, and that wouldn't make for a very good marriage. So right now I'm keeping them all inside.
Well, I'm a beekeeper too, and I have a lot of sarracenias outside and Venus flytraps, and they never catch any of my bees. Every other bug in the world, but no bees. So reassure her.
I know that when I open up some of my traps at the end of the year, yeah, there are bees in there, but they're usually yellow jackets. majority of the time they're yellow jackets or just, flies of any size, shape, or kind.
Okay, thank you, because I know she sets traps out for different hornets and so on. So I appreciate all the good responses I can give to her.
Yeah, yellow jackets are wasps, so they're attracted to
Right.
meat, protein. So yeah, they're not in the same category as a honeybee.
Right. Yeah, exactly. And they're predatory towards honeybees.
Yeah.
I do get some
Thank
carpenter bees in my Sarracenia sometimes in the summers. They'll get drunk on the nectar and then fall in. But then they just cut themselves a little hole at the bottom of the pitcher. All day long, they just go in circles. So if you're worried about those, don't worry about that.
So what, they get drunk and then they sober up and they go up to the top and get some more?
Yeah, they just cut a perfect little circle at the bottom and then they crawl through it. So all my pitcher plants have holes in them, usually in the summers.
Well, hi guys. Hope it's okay if I cut in. My name is Yaron. I used to live in New Jersey and Pennsylvania, and I left the country, and now I live in Israel. And I brought a bit of home with me. I have here a bunch of Sarracenias that I grow here, amongst other things such as sundews, some nepenthes, and a few Venus flytraps. And I'm happy to be a part of this group.
Awesome. Thank you.
Thanks. Welcome.
Hi. And here it's nighttime at the moment, so the lighting is very poor, but I turned on some spotlights here that I have, but so as I said, it's kind of nice.
How is the climate there in Israel for growing them? Are you able to grow them outside year round, or do they have to be?
Well, basically, yes, I do grow them outside. I do have them under a 50% shade cloth because the sun can be very harsh out here. And when we have the desert winds coming in from the east here during spring and autumn, I do have some misters to supplement humidity and bring it up to about 50%. But during the summer, the Sarracenia are very happy here. They bloom, they have great coloration, and they show wonderful pictures.
There's a question in the chat actually from Doris about carnivorous plants not eating their pollinators. I think there's probably a lot more knowledgeable people on this subject here, but generally my observation is that this is a kind of general observation that people have had. Mostly it stems from the idea that Sarracenia, that Venus flytraps, they send up their flowers pretty early on in the year. It's actually before the traps fully develop. And so the idea, from an evolutionary standpoint, is that the plant doesn't want to eat its pollinator. And so it would send up the flower before the traps are actually active. That being said, though, I don't know if there are any specialized pollinators for Sarracenia or for Venus flytraps. I feel like the answer in biology is always probably yes, but I definitely can't give you a definitive answer on that. I don't know if anyone else has any thoughts.
I have this. I actually just was looking at this book. It's a field guide to wildflowers of the eastern United States, and it does have Sarracenia purpurea in there, and it says pollination is bees. It's got a little picture of a bee. So that's not like a super in-depth answer, but I literally just was looking at this book this morning, trying to find something. And so yeah.
Any other questions? Just, anything from very beginner to very advanced, we'll take it all. Or if you want to just say hello, please do.
Doris is asking in the chat about the International Carnivorous Plant Society conference. If anybody's gone in the past, what it was like, and if anyone's planning on going.
Well, I have been to the recent one in Japan, which was quite amazing. We had wonderful field trips, 3 days of lectures that included the simultaneous translation to English, and that offered a very awkward moments in certain translations. The lectures themselves were quite well, ranging anything from biochemistry of plants to conservation and many subjects. Some of the movies are coming on now online on YouTube on the International Carnivorous Plant Society their channel, and I will be attending this year in Austria in May as well. And there won't be field trips such as the ones that were in Japan to in situ sites, but we will be visiting over there several growers. We will be seeing some grow houses in the museum or in the botanical garden in the area. I hope that answers the question.
Yes, thank you.
Now it comes down to saving the money to get to Austria.
Well, the hotels are very expensive there, so that's more of an issue.
Kevin, you said you'd bought some things from Diflora before. What have you bought from them so far? Are they like, is he putting out unique cultivars of fly traps or anything like that, or is it known quantities of stuff that we've seen before?
Yeah, I actually bought from them just because I was very impressed with their selection. They had things that I had never seen before in the States. We're talking different, rare cultivars of Venus flytraps. We're talking Drosera species like Drosera curvipes. I don't even know how to say it. But, it's like these recently discovered Drosera. I think he also has tuberous sundews as well. Things that just traditionally have not been easy to find in the States. He has some pretty nice Nepenthes hybrids as well. You know how I go for the Ambularia / Mirabilis globosa hybrids, and he had a bunch of those. So when I ordered them, I was actually really impressed with the quality. I mean, you get like you heard from him today, he had they basically come out of tissue culture. And so when you buy, say, Venus flytrap, you're not buying one plant, you're actually buying more like 50 plants. Very small, but it's always great when you get a huge clump of them. And so I think he's got continuously just like more and more rare things. And so I think it's great that we'll be able to order from him directly.
Yeah, I'll definitely have to be keeping a close eye on what they're putting out for sure, because I definitely think that's interesting. Are you getting his stuff like in the flask still, or is it just sort of like with Stuba where it's bare root but still kind of like fresh out of the the flask and whatnot?
Yeah, so I was hoping to actually ask him a little bit about that, and he talked about his acclimatization process. So when he shipped them to me, they were actually, I think, either in Ziploc bags or maybe he had a more elaborate shipping structure, but they were not in the flasks anymore. That being said, they were still very tender, and so for the Venus flytraps, I basically hardened them off in my Nepenthes greenhouse over the period of a couple of weeks. So there is still a process, but they're not nearly as tender as just fresh out of tissue culture.
Okay, yeah, no, that's good to know. I mean, that's kind of what I'm used to seeing with like BCP or whatever, where you get the stuff in and it's very sensitive still, but you do get a lot of material typically from him.
Yeah, that describes that pretty well. If there's no other comments, I think we might leave it at that. We have, as you saw, I posted in the chat as well just a couple of minutes ago, we have a whole bunch of events lined up for this year. We will send you more information about our field trip. It really should be an amazing one. I mean, people have been talking about going to the Pine Barrens for a very, very long time. So now is finally your chance. We'll also have a couple other in-person meetings that don't involve getting as muddy later in the year, and we have all those pop-up events. So if you want to sell plants at our tables or just simply educate people about carnivorous plants, please feel free to reach out to us. With that, I will wish everyone a happy rest of your weekend and hope to see you in person soon.
Thank you very much. Have a wonderful weekend.